Chapter 18, the circulatory system, blood. There's many myths about blood, that it was a mysterious vital force. In ancient times they drained bad blood for medical reasons, and hereditary traits were once thought to be transmitted by blood. The expected learning outcomes. Describe the functions and major components of the circulatory system. Describe the components and physical properties of blood. Describe the composition of blood plasma. Explain the significance of blood viscosity and osmolarity. And describe in general how blood is produced. Functions of the circulatory system. The circulatory system has three main functions: to transport, to protect, and to regulate. It transports oxygen, carbon dioxide, nutrients, waste, hormones, and stem cells. Protection it offers is that the blood plays several roles in inflammation, a mechanism for limiting the spread of infection. White blood cells destroy microorganisms and cancer cells. Antibodies and other blood proteins neutralize toxins and help to destroy pathogens. And platelets secrete factors that initiate blood clotting and other processes for minimizing blood loss. Regulation, it absorbs or gives off fluid under certain conditions. The blood capillaries help to stabilize fluid distribution in the body. By buffering acids and bases, blood proteins help to stabilize the pH of the extracellular fluids. And shifts in blood flow help to regulate body temperature by routing blood to the skin for heat loss or retaining it deeper in the body to conserve heat. The circulatory system consists of the heart, blood vessels, and blood. Cardiovascular system refers only to the heart and blood vessels. Hematology is the study of blood. Components and general properties of blood. Adults have 4 to 6 liters of blood. Blood is a liquid connective tissue consisting of cells and extracellular matrix. Plasma is the matrix of blood, and it is clear or light yellow fluid. The formed elements of blood are the blood cells and cell fragments such as red blood cells, white blood cells, and platelets. Notice this blood smear. Erythrocytes are red blood cells. Neutrophils are white blood cells. And platelets are parts of the cells or cell fragments from it. There's 7 kinds of formed elements. Erythrocytes, or red blood cells. Platelets, which are cell fragments from a special cell in bone marrow. And the other 5 formed elements are leukocytes, or white blood cells. Granulocytes are white blood cells with granules, and those are the neutrophils, eosinophils, basophils. Agranulocytes is without granules, lymphocytes, monocytes. Take a look at this picture. The granulated ones, eosinophil, neutrophil, and basophil, have granules all throughout them. Eosinophil is typically double-lobed, and neutrophil has several lobes or multi-lobes. The basophil is just granules all throughout. Monocytes have a lobed or kidney-shaped nucleus, and lymphocytes have a rounded nucleus that take up a great majority of the cell. Monocytes and lymphocytes do not have granules. Neutrophils are phagocytic cells of the nervous system, and they're often the first to arrive after an inflammatory response is initiated. Eosinophils are often elevated in multicellular parasitic infections. Lymphocytes are involved in immunity. B cells produce antibodies that neutralize foreign objects, and T cells produce cytokines that kill pathogen-infected cells. Basophils contain histamine, so they're important in parasitic and allergic reactions, and monocytes often serve to replenish macrophages. Half of them are stored in the spleen. A hematocrit is a centrifuge blood to separate the components. Erythrocytes are the heaviest, and they settle on the bottom, and they're about 37 to 52% of the total volume. The bottom is shown as red. White blood cells and platelets are only about 1%, and they're called a buffy coat, and they're located right above that erythrocytes. And then the remainder of the volume is the plasma, which is a clear or yellowy type liquid. It's about 47 to 63%, and it's a mixture of water, proteins, nutrients, electrolytes, nitrogenous waste, hormones, and gases. So plasma is the liquid portion of the blood, and the serum is the remaining fluid when blood clots and the solids are removed. And serum is identical to the plasma, except it does not have the fibrinogen. There's 3 categories of plasma proteins: the albumins, the globulins, and the fibrinogen. The albumins are the smallest and most abundant, and they contribute to the viscosity and osmolarity. They influence blood pressure flow and fluid balance. The globulins are antibodies, and they provide immune system functions, and there's alpha, beta, and gamma. The beta, which is transferrin, transports iron, and gamma antibodies combat pathogens. Fibrinogen is the precursor of fibrin threads that help form blood clots. Plasma proteins are formed by the liver except for the globulins, which is produced by plasma cells. Nitrogenous compounds are free amino acids, which are from dietary protein or tissue breakdown, and the waste, called urea, are the toxic end products of catabolism or amino acid catabolism, and is normally removed by the kidneys. Since many of these proteins are formed by the liver, liver cancer or hepatitis can result in impaired blood clotting. Blood plasma includes nutrients such as glucose, vitamins, fats, cholesterols, phospholipids, and minerals. It also contains dissolved oxygen, carbon dioxide, and nitrogen. Nitrogen normally has a non-physiological role in the body. And all blood plasma also contains electrolytes. Sodium makes up 90% of the plasma cations, and it is important for blood osmolarity, so people with high blood pressure are often advised to limit sodium intake. Viscosity is the resistance of a fluid to flow, the stickiness resulting from the cohesion of its particles, and whole blood is 4.5 to 5.5 times as viscous or stickier than water. Plasma is twice the times as viscous or sticky as water, and that's important in circulatory function. Osmolarity of blood is the total molarity of those dissolved particles that cannot pass through the blood vessel wall, and if it's too high, the osmolarity absorbs too much water, increasing blood pressure, and if it's too low, too much water stays in the tissue. Blood pressure drops and edema occurs. The best osmolarity, or the optimum osmolarity, is achieved by the body's regulation of sodium ions, proteins, and red blood cells. Kwashiorkor is a condition that occurs when there's a protein deficiency, and so you get edema or fluid in the tissue. What happens is usually blood rushes into the capillary bed, and the hydrostatic pressure builds and pushes fluid out of the vessel. When a blood pushes back into a vein, the colloid osmotic pressure brings fluid back into the vessel. In kwashiorkor, there's a protein deficiency, so it can't make albumin and other blood proteins, and without albumin, you get fluid in tissue and edema. The tissue is so flooded that it is hard to get the nutrients in, and the tissue will fall off. Blood is produced every day. Adults produce 400 billion platelets, 200 billion red blood cells, and 10 billion white blood cells every day. Hemopoiesis is the production of blood, especially its formed elements. Hemopoietic tissues produce blood cells. The yolk sac produces stem cells for first blood cells, and it's colonized fetal bone marrow, liver, spleen, and thymus. And the liver stops producing blood cells at birth, but the spleen remains involved with lymphocyte production. Red bone marrow produces all 7 formed elements. Remember, that is the platelets, the red blood cells, and all 5 of the leukocytes. Pluripotent stem cells, formerly called hemopoietic stem cells, in the bone marrow can become any of the cells. That's called myeloid hemopoiesis. Basically, it becomes the colony-forming unit, and the colony-forming unit has a receptor for EPO. The cell binds to the EPO, becomes a uroblast, leaves the bone marrow, and becomes a erythrocyte. Colony-forming units are specialized stem cells only producing one class of formed element of blood, and lymphoid hemopoiesis is blood formation in the lymphatic organs. Erythrocytes, red blood cells. Expected learning outcome, discuss the structure and function of them, discuss the structure and function of hemoglobin, state and define some clinical measurements of red blood cells and hemoglobin quantities, describe the life cycle, and describe types, causes, and effects of red blood cell excesses and deficiencies. They have two principal functions. They carry oxygen from lungs to cell tissues, and they pick up carbon dioxide from tissues and brings it to the lungs. Insufficient red blood cells may kill in minutes due to lack of oxygen to tissues. They are disc-shaped cell with a thick rim, and they lose nearly all of their organelles during development. They lack a nucleus or mitochondria, so they have anaerobic fermentation to produce ATP, and they lack the nucleus and DNA, so they have no protein synthesis or mitosis. The lack of mitochondria ensures that they deliver oxygen without wasting or using any of the oxygen, and the lack of DNA means that they cannot make repairs to themselves, so they have a short lifespan of 120 days. Blood type is determined by the surface glycoprotein and glycolipids. Red blood cells have a cytoskeleton, and it's made up of protein spectrin and actin, which give the membrane durability and resilience. The red blood cell stretches and bends as it squeezes through small capillaries, but the spectrin and actin eventually ages and becomes compromised and bursts open. The spleen has the smallest capillaries, so it happens most frequently here, and is called the red blood cell or erythrocyte graveyard. Once the red blood cell bursts, macrophages come to eat them. Red blood cells are very important in gas transport, and that's because they have an increased surface area to volume ratio because they've lost all of their organelles while they've been maturing. So this increases the diffusion rate of substances, allowing them to transport things better. 33% of the cytoplasm is hemoglobin. There's 280 million hemoglobin molecules on one red blood cell. Oxygen delivers to tissue, and the CO2 transports to lungs. And although red blood cells are especially known for oxygen transport, it also aids in the transport of carbon dioxide and the buffering of blood pH. The cytoplasm also contains an enzyme, carbonic anhydrase, that catalyzes the reaction carbon dioxide plus water to H2CO. So each hemoglobin molecule consists of 4 protein chains, which are called globins, and 4 heme groups. And heme groups are non-protein moieties that bind oxygen to ferrous iron, or Fe2, at its center. HbA is normal hemoglobin, HbF is fetal hemoglobin, and HbS is sickle cell hemoglobin. Red blood cell count and hemoglobin concentration indicate the amount of oxygen blood can carry. The hematocrit, or the packed cell volume, is the percentage of whole blood volume composed of red blood cells, and in men it's slightly higher, from 42 to 52%. In women, it's from 37 to 48. The hemoglobin concentration of whole blood should be for men between 13 to 18 grams per deciliter and women 12 to 16. And red blood cells for men is 4.6 to 6.2 million per microliter, and women 4.2 to 5.4. Values are lower in women because women don't make as many androgens, and androgens would stimulate the red blood cell production. Women also lose a lot of their erythrocytes during their periods. And hematocrit is inversely proportional to percentage of fat. Since women have a higher fat percentage, lower hematocrit. In men, blood clots faster and skin has fewer blood vessels than in women. From an evolutionary standpoint, this might be because men fought more than women did, so they suffered more injuries, so they kept blood flow away from the skin where they would be hurt. Erythrocyte production begins with a pluripotent or hemopoietic cell that goes to a colony-forming unit, which has receptors for erythropoietin, and the EPO, or erythropoietin, stimulates the colony-forming unit to transform into erythroblast. The erythroblasts multiply and synthesize hemoglobin. When it's done, the nucleus shrivels and is discharged, and it's called a reticulocyte. And it basically looks very similar to a basophil, but it has a network composed of ribosome clusters. And about 2.5 million of these are produced per second, with an average lifespan of about 120 days. Development of them takes 3 to 5 days. In those 3 to 5 days, it's reducing in cell size, increasing in cell number, synthesizing hemoglobin, and losing its nucleus. The first committed cell, which is the eCFU or erythrocyte colony forming unit, is the one that has receptors for erythropoietin or EPO from the kidneys. The erythroblasts multiply and synthesize hemoglobin, and then the nucleus is discarded and forms a reticulocyte. And about 0.5% to 1.5% of circulating red blood cells are reticulocytes. This can rise in certain circumstances, such as blood loss, for example. This accelerates erythropoiesis and leads to an increasing number of reticulocytes in circulation, as if the bone marrow is in such a hurry to replenish the lost RBCs that it releases many developing RBCs into circulation a little early. Since we absorb only a fraction of the iron in our food, we must consume 5 to 20 milligrams per day to take care of these losses. A pregnant woman needs 20 to 48 milligrams. Iron is a key nutritional requirement, and it's lost daily through urine, feces, and bleeding. Men lose about 0.9 milligrams per day and women 1.7. But it's such a low absorption rate. That's why we need to have so much more intake. Iron metabolism. There's 2 types, ferric, Fe3+, and ferrous, Fe2+. The stomach acid converts Fe3+ to absorbable Fe2. Gastroferritin binds the Fe2 and transports it to small intestine. It's absorbed into blood and binds to transferrin for transport to bone marrow, liver, and other tissues. In the bone marrow, it's used hemoglobin, sulfur-myoglobin, and all cells use for cytochromes and mitochondria. In the liver, apoferritin binds to create ferritin for storage. Only the ferrous, Fe2, can be absorbed by the small intestines, which is why So stomach acid, as a review, converts most of the ferric to ferrous, because that's the only kind that can be absorbed by small intestine. Gastroferritin then binds to the ferrous and transports it to the small intestine. Here it's absorbed to the blood, binds to a plasma protein called transferrin, and travels to the bone marrow, liver, and other tissues. Bone marrow uses iron for hemoglobin synthesis. The muscle uses it to make oxygen-storing protein myoglobin, and nearly all cells use it to make electron transport molecules called cytochromes in their mitochondria. The liver binds surplus iron to a protein called apoferritin, forming an iron storage complex called ferritin, which releases more ferrous, Fe2, when needed. So step by step, ferric converted to ferrous by HCl. Gastroferritin binds to ferrous, transports to small intestine, releases it, and is absorbed in the blood. Transferrin binds and escorts ferrous in blood to liver and bone marrow, and in the liver, it is passed off to apoferritin to be stored as ferritin, and also goes to bone marrow for hemoglobin. Iron metabolism. Vitamin B12 and folic acid are responsible for rapid cell division and DNA synthesis that occurs in erythropoiesis. Vitamin C and copper are responsible as cofactors for enzymes synthesizing hemoglobin. Copper is transported in the blood by an alpha globulin called ceruloplasmin. Erythrocyte homeostasis is a negative feedback control. Drop in red blood cell count causes hypoxemia. Kidney production erythropoietin stimulates bone marrow. Red blood cell count increases in 3 to 4 days. And the stimuli for increasing erythropoiesis are low levels of hypoxemia, a high altitude, increase in exercise, loss of lung tissue, and emphysema. So going counterclockwise starting at noon, there is an inadequate O2 transport or hypoxemia. That's sensed by the liver and kidneys, which secrete the erythropoietin, which stimulates the red bone marrow, which accelerates erythropoiesis, which increases the red blood cell count, which increases the O2 transport. The heme is pigment converted to biliverdin. The biliverdin converts to bilirubin, which is released into the blood plasma, into the kidneys, which turns the urine yellow. The liver removes the bilirubin and secretes into bile. Bile is concentrated in the gallbladder, released into small intestine, and bacteria interact with it, creating urobilinogen, which gives us brown feces. Erythrocyte death and disposal starts in the small intestine, where the nutrients are absorbed. Then erythropoiesis is in the red bone marrow. The erythrocytes circulate for 120 days. Then the expired erythrocytes break up in the liver and the spleen. Some of the cell fragments are phagocytized, but the hemoglobin is degraded into heme and globin. Globin is hydrolyzed to free amino acids, and the heme is broken down into biliverdin and iron. The macrophages remove the Fe2 from heme and releases into blood. The heme is broken into biliverdin and iron. The iron is stored or reused or lost. The biliverdin turns into bilirubin. Bilirubin goes into blood, binds to albumin, transports to liver, stored in bile, and released into the intestine or bile. Bacteria converts into urobilinogen, giving feces a brown color. Disorders of red blood cells. Polycythemia is an excess of red blood cells. There's primary and secondary. Primary is typically from cancer in red bone marrow. Secondary is from dehydration, emphysema, high altitude, or physical conditioning. Primary can lead to red blood count cells as high as 11 million per microliter with an 80% hematocrit. And secondary can lead up to 8 million. And polycythemia can increase blood volume, pressure, viscosity, leading to embolism, stroke, or heart failure. Blood volume can double in primary and cause the circulatory system to become engorged. Viscosity may rise to 3 times normal, circulation is poor, capillaries are clogged, and the heart is strained. Deadly consequences of emphysema and other lung diseases are in part to polycythemia. Anemia falls into 3 categories. There's inadequate erythropoiesis or hemoglobin synthesis, hemorrhagic anemias, and hemolytic. Hemolytic anemias come from red blood cell destruction that can be caused by penicillin allergies or other drug reactions, by poisoning such as mushroom toxins, snake and spider venoms, or parasitic infections like malaria, or even hereditary defects like sickle cell or thalassemia, and blood type incompatibilities like hemolytic disease of the newborn or transfusion reactions. Hemorrhage anemias come from bleeding. And inadequate erythropoiesis or hemoglobin synthesis can be caused by kidney failure, iron deficiency anemia, or pernicious anemia. Pernicious anemia is especially dangerous in elderly people. It's an autoimmune disease in which antibodies destroy stomach tissue. Without stomach tissue, we do not have the intrinsic factor needed to absorb the iron or the vitamin B12, This is also a problem in people with gastric bypass because they had stomach tissue removed, so they do not have the intrinsic factor. This type of anemia is treatable with B12 injections or oral B12. Anemia has 3 potential consequences. There's tissue hypoxia and necrosis, where the patient is lethargic, there's shortness of breath, and there's necrosis of brain, heart, or kidney. Blood osmolarity is reduced, producing tissue edema, and blood viscosity is low, so heart races, pressure drops, cardiac failure may occur. The blood osmolarity is reduced because more fluid transfers from the bloodstream to the intracellular spaces, resulting in edema. Viscosity reduces because the blood puts up so little resistance to flow, the heart beats faster than normal, and cardiac failure may ensue. Blood pressure also drops. Sickle cell disease, from a hereditary standpoint, it affects mostly people of African descent because something in the sickle cell actually helps the people survive malaria. It's caused by a recessive allele that modifies the structure, differs only on the 6th amino side. The HBS does not bind oxygen well. The red blood cells become rigid, sticky, and pointy, almost crescent-shaped. They clump together and block small blood vessels, and it can lead to kidney or heart failure, stroke, rheumatism, or paralysis. Blood types. Antigens are the molecules on surface of cell membrane that are unique to the individual. a foreign antigen and generates an immune response. Antibodies are proteins secreted by plasma cells, and it's part of the immune response. Forms antigen-antibody complexes, and agglutinogen is an antibody molecule binding to antigens, causes clumping of red blood cells. Red blood cell antigens are called agglutinogens, and there's A and B, and it's found in plasma. A blood type A person has the A antigen. B person has a B antigen. AB person has A and B, and O has neither. If you're blood typing and you put anti-A into type A, you will get clumping. You will also get clumping when you put it into AB, since AB also has A antigens. If you put anti-B, you will get clumping in B and AB, again because AB has anti-B antigens. You will not get clumping in type O because O has neither A or B. You do not form antibodies against your antigen. Agglutination. Each antibody can attach to several foreign antigens on several different red blood cells at the same time. And this is what is responsible for mismatched transfusion reaction. Agglutinated red blood cells block small blood vessels hemolyze and release their hemoglobin, causing blockage of the kidney tubules and acute renal failure. Take this for example. A type A or type B person has type B antigens. A type A donor donates. The recipient's plasma does not recognize it, so it attacks the type A, causing it to agglutinate, and the red blood cells block small vessels. Type O is the most common blood type, and it lacks red blood cell antigens. There's no A or B. But the donor's plasma may have both antibodies against anti-A and anti-B. So by packing the cells and having minimal plasma, that can be avoided. Type AB is the rarest. There's no anti-A nor anti-B, so it can receive anyone's. If an Rh mother is pregnant with an Rh negative mother is pregnant with an Rh positive baby, her body may begin to make anti-Rh or anti-D antibodies, which may affect the infant. But it can be prevented with RhoGAM. So what happens in hemolytic disease of the newborn? If a Rh woman gives birth to or miscarries an Rh child, when the child is born, there is blood mixing. So the woman's blood begins to create anti-D antibodies. These anti-D antibodies then are present in her bloodstream. So in a subsequent pregnancy, if her child is Rh positive, her anti-D antibodies may attack it. If this happens, it can cause hemolytic disease of the newborn, which can cause high bilirubin levels, and it can cause high anemia, and it can also cause toxic brain syndrome, and because it lyses fetal blood and severe anemia, and it can't be prevented by giving mom RhoGAM.